The overall term of epigenetics refers to changes in gene expression. So this can come about because of a few different things, which can influence which genes in our genotype are silenced, which genes in our genotype are activated, and then this will in turn give rise to a phenotype, so structural and functional proteins being made from genes which lead to particular physical observable traits or behavioural traits and things like that in an organism. So this can be, these gene changes can occur from organism to organism, or even when we look at cells, from cell to cell, gene expression can have an influence on types of cells becoming certain cells becoming particular types of cells and so on. So I'll talk about that in a minute. But the way that we see gene expression being altered comes about from a couple of methods previously mentioned, things like DNA methylation, so the addition of methyl groups to cytosine bases in our DNA in order to silence genes. We also see chromatin remodeling, so the alteration of our chromatin, which sort of largely comes about because of histone acetylation. We're adding acetyl groups to histone proteins, which our DNAs are wrapped around. We'll mess around with the charges, and we'll see the spreading out of histones and the exposure of genes, which will be activated and then possibly expressed, because they can be accessed by RNA. All right. So if we look at a cellular level, the impact or the influence that these effects on gene expression can have, initially in an early sort of embryonic stage, we start out with a whole bunch of stem cells. So this is going to be my stem cell here. So stem cells have the potential to become any type of cell because of the activating or silencing, expressing or silencing of genes within its DNA, which is found in the nucleus. We know that all cells have the same set of DNA. They have different phenotypes, different appearances and functions, because some genes are expressed, some are silenced. So that gives rise to differences. So if we have a stem cell, what's going to occur is epigenetic factors. So factors like the histone acetylation, the DNA methylation are going to lead to phenotypical change because of particular genes being expressed or silenced. So let's say that this stem cell, because of these epigenetic factors, it's going to look the same, but let's say that this was a liver cell. So these epigenetic factors influence the stem cell, which has not got a role yet, has not got a particular cellular function. It can become any type of cell, but the genes that have been activated have led to it becoming a liver cell. From this point on, the liver cell will then retain the certain activated factors and the certain silence factors or silence genes, which means that through the process of mitosis, so the copying, creating clones of these liver cells through cell division, we end up with more liver cells. So after a stem cell has undergone particular epigenetic factors to become a certain type of cell, such as liver cell, blood cell, whatever, then those cells undergo mitosis and those same genes stay activated and silenced, and we give rise to more liver cells with the phenotype of liver cells, so expression of the structure and function and appearance of liver cells. So this can happen also in sperm cells. This is going to be my sperm cell, like that. I'm sure again that's a pretty ordinary sperm cell. Whoops. The tail should be a little bit thinner. That'll do. So we've got our sperm cell, and we're also going to have an egg cell. Now, egg cell is just going to look like that. All right. So the sperm cell and the egg cell will come together through the process which we refer to as fertilization in order to create a zygote. So a zygote is the first cell of what will be an embryo and eventually grow into offspring, a baby in humans. The zygote is the very first time where a father's sperm cell and mother's egg cell combine for fertilization. So this is a single cell which is going to eventually become this multicellular human organism. Let's say, for example, at the time of fertilization, the sperm cell had X, Y, and Z genes, to be very, very generic. X, Y, and Z genes silenced. And the egg cell had these genes called A, B, and C genes silenced. So if those particular genes are silenced, among others being activated at the time of fertilization, then in this zygote, those genes are going to stay activated or silenced. So we see A, B, C, X, Y, Z silenced. So this is what helps give rise to a particular phenotype rising from the genotype because of these epigenetic factors that are switched on or turned off, genes that are silenced or activated at the time of fertilization. So that's why certain genetic factors are passed on from parent to progeny, and why, sort of leading down the fact of different alleles, which can be dominant or recessive. That's a conversation for another time. We can sometimes see appearances in children that weren't necessarily seen in the parents because of different genes being activated that weren't activated when both of their parents were inseminated, right back at the very beginning, at the zygote stage. So the last little point that I want to make is all of this stuff is not necessarily beautifully controlled without being influenced by external things such as environmental factors. So we can see genes, these epigenetics, being influenced for good or bad by things like diet. Also a very big one is drug use, specifically, or especially rather, during pregnancy. So things that should be activated, genes that should be activated, genes that should be silenced, could be messed around with due to drug use during pregnancy, which can give rise to problems with a fetus and eventually a baby. Another point to make as well, I guess stemming on from that, is epigenetic factors. If they go wrong, if environmental factors come into play in a negative sense, we can get disease. So that comes about, like I sort of mentioned just before, through incorrect, what we'd say, activation / silencing. So if the wrong genes are activated and the wrong genes are silenced, then we're not going to develop particular cells or limbs of a baby or organs and things like we would have desired. If the right genes were switched on, then the right genes were switched off. So that's a rundown to epigenetics.